So up next, we have Dr. Craig Campbell and Bonnie, who are going to be talking about the treatment landscape and clinical trials. And so Dr. Campbell is going to take us through the treatment landscape, and then Bonnie's going to talk a little bit about the clinical trial concierge position with the Neuromuscular Disease Network for Canada. And hopefully we'll have enough time that I'd like to show you our clinical trial tracker tool that we had developed. So I will pass it over to Dr. Campbell. Thank you.
Thanks, Nicola. That's for your video or your show? Yeah, great. Okay, thanks so much. Move that down a little bit. So we're going to walk through the treatment landscape for DMD, and then I'm going to hand it over to Bonnie near the end of this. Bonnie also works with us a very important role in helping to coordinate some of the clinical trial activities across Canada, and of course Nicola is a constant partner in many of the activities we're doing as a clinical trial network, which I'm going to show you a little bit about as we get further into this. So what I'm going to try and touch on is three things, or really two things, but then put some examples in, really some foundations for understanding emerging therapies. And I'm going to try and frame that in terms of categories of emerging therapies. We've got a great number of industry partners here who will have the better part of the agenda tomorrow. And I feel like they're going to be the ones to show you what's specifically happening in their clinical trial program. So you're going to see I'm going to stay fairly superficial on that, which may be different than a talk you're used to on kind of emerging therapies where, we kind of go into, oh, this is in phase 1 studies and all that. I just thought the way the agenda works here for tomorrow is you'll be able to really have some sightlines into what some of the companies are doing. So I'm going to take it, I think, or try and take it in more of a conceptual and categorical way. Again, I have a great many disclosures. We're a very active clinical trial site here in London, and work with many companies. And again, I, despite doing consulting analysis, work with many of these companies and a site PI for many studies. I don't take any personal money for that other than for data safety monitoring committees, as I mentioned earlier. So again, I'm not going to talk about treatment of symptoms and complications. I think just to put this in context, that's a lot of the things we've been talking about in the talks, the lectures that have come around the standards of care. So, things like steroid use or bisphosphonate use, or, orthopedic things and all that. So really what I'm going to focus on is the emerging therapies. And again, I just put this in as a reminder because I always want to ground things on what matters in our care approach. So I'm going to start with the foundation for emerging disease-modifying therapies, and I use this description specifically because, again, I want to be clear that at this point in time, we actually don't have any approved disease-modifying therapies in Canada. Steroids would be our treatment that we use, and for good reason and very effective. But what I'm about to show really are things that are in clinical trial development largely in Canada and mostly around the world. But there are a few of these therapies, and I'll try and remember to point those out when we get to those spots where, particularly in the US, but also in one situation in Europe, there are some treatments that are available by prescription. So to start understanding the the treatments for Duchenne, or the emerging treatments, the treatments that are in clinical trial development, one really needs to understand genetics. So we're going to go back and do a little bit of sort of genetics 101, actually, and I don't know if I'd be able to move without a mic, but maybe. So what if I want to start with is just reviewing sort of DNA, RNA, and protein, and you'll, oh, perfect. And you'll maybe appreciate these documents or these diagrams from other things you've read. I mean, we're in a genetic era, and so people are often thinking about or reading about DNA, but DNA is sort of the blueprint for all your genetic activity. And on these long helix-like chemicals that is the blueprint for our body, there's one of these areas on the X chromosome that codes for the dystrophin protein, which I'm going to show here. But there's a step in between the DNA and the production of this protein, which is represented, say, by these bead-like diagram down here that says amino acids. These amino acids line up, and then once they're put together in the right sequence, they fold up and they make this, this dystrophin protein that we talk so much about, and I'll come back to this. But there's a step in the middle that's actually really important to understand for many of the emerging therapies in DMD, and that's this RNA. And this is a mirror image to the DNA, as you can see, but it's sort of a single strand element, and it exists to take the blueprint to the actual building. And so a lot of our therapies that are emerging, like exon skipping or nonsense read-through, are not so much DNA-based modifications or interventions, they're RNA modifications. And so I'm going to call those or refer to those as genetic-based therapies rather than gene therapy. And I'll give you some examples to put that in context. So once the dystrophin gene makes an mRNA, this mRNA hangs around in the cell for a little bit and produces dystrophin protein. It puts these building blocks together, the bricks and mortar of the dystrophin gene, and it produces this this protein here. And just wanted to situate this for you as a whole muscle, a muscle fiber here. And then if you really drill down on this muscle fiber, you see an individual muscle cell or a myofiber. And that individual muscle cell has a membrane. It's contained in a lipid or fatty cell wall. And dystrophin is really, really critical in attaching the inside workings of the muscle cell to the cell membrane through a series of other proteins, which we're not going to talk about, but are actually each of these proteins represented here, which I haven't named, they actually are problematic in other muscle diseases, but we're obviously here to talk about Duchenne. So this dystrophin protein acts to basically anchor the outside of the cell to the inside of the cell. And you can see this architecture so important because you need all these muscle fibers sort of working together. And if you can imagine, just millions and millions of these dystrophin proteins providing that scaffolding for us in the muscle. So back to this. So the dystrophin gene is actually one of the largest genes in the body. And as such, like anything in the cellular machinery, if you're very big, you're apt to have, just spontaneously have some challenges sometimes. And so dystrophin does undergo mutations. The gene has mistakes happen to it. And that happens about 30% of the time. And we categorize those mutations into different types. So this, and these circle charts sort of show that. This shows that most of the mutations, most of the problems that happen with the dystrophin gene that cause it not to produce dystrophin very well are large deletions or duplications in the gene, okay? So they can be picked up on a certain type of genetic test. So when you hear about people saying, oh, I have a deletion of, 48 to 50, that's we're talking about this big category. The next biggest group is point mutations. So if you can go back to that gene diagram, we're talking about just one or a small number of those elements that code for a protein missing, and when that's missing or not the right sequence, we call those a point mutation or a small mutation. And out of that, and then there's a number of other smaller kinds of mutations or problems that can happen in the gene. Out of this 30%, about half of those are nonsense mutations. And that's an important type of mutation for a certain therapy called Adlorin or nonsense read-through. And a nonsense mutation basically tells the cell machinery to just stop right where you are. It tricks the cell machinery that's trying to produce dystrophin. It gets to that point and it has a stop signal, like a stop sign that says, we're done here. And when you hit that, the body stops producing dystrophin. And so one of the therapies that's in evolution and actually approved in a number of countries tries to trick the cell to keep reading through that. And that is called Adlorin, that drug. As I mentioned, the dystrophin gene is a huge gene. It has 79 coding areas or sections that make up the whole gene. That's very big compared to many genes in the body. And they fit together in this sort of puzzle piece way. You can see, 56 matches up with 57. And so if you can imagine going back to my slide, if you have a big deletion in an area, if you're missing one of these, if in the course of development, one of these sections has gone missing, then you can imagine that, you can see 57 doesn't line up with 59. They have different, what we call reading frames. And so if you all of a sudden get rid of 58, 57 can't keep going and keep reading or keep producing dystrophin. So again, it stops right there and says, uh-oh, I'm in trouble. I don't know what to do anymore. And we can capitalize on these circles, by the way, are just what we call hotspots, where many of the mutations are. A high proportion of boys with Duchenne have mutations in these areas, which is why some of the targets have been set. Particularly, we talk about exon 51 skipping. You'll hear that. That's because really that region is the most common area where there's a mutation. But you can see that if when we talk about exon skipping, which I'll show you later, we are using this puzzle piece sequencing for the dystrophin gene to specifically skip over areas so that we can line up these this reading frame again. And so again, it's a question I often get. People say, well, why do you just pick 51, or why wouldn't you just make one for my deletion? It really has to do with doing this intricate boutique work that many of these companies are doing to find the exons which end up allowing us to create the reading frame again as often as possible in as many boys as possible. And so I show dystrophin here again in a slightly different schema. It's here, but again showing that it has a major structural property in the muscle cell, but it also has many other functions. And I list these not to be overly complicated, but mostly to say that many of the therapies that have been tried over time in Duchenne muscular dystrophy actually try and get at some of these impacts that that dystrophin absence causes. So for example, dystrophin, again, a structural protein here, but when I say an impact on satellite cells, one of the unique things we're learning about, and it comes from Michael Rudnicki's lab, and I think we have someone from Satellos here, is that dystrophin actually helps the muscle stem cells to get in the right polarity so that they can divide and replicate. And so again, we wouldn't have necessarily, kind of thought or imagined that dystrophin might play a role in all these little satellite cells that aren't actually even at that point contributing to the architecture of the muscle. But in fact, it does. And when stem cells can't orient themselves properly, they don't reproduce. And if you don't reproduce, those stem cells don't incorporate themselves back into the injured muscle, and that's how we naturally correct muscle injury. But fundamentally, the thing you can walk away from, despite all these other things that dystrophin helps contribute to, is if you don't anchor the inside of the muscle cell to the outside of the muscle cell, as you undergo muscle contraction over time, there's muscle injury, then the muscle cell, it can't recover well from that, and there's muscle breakdown and mechanical strain on the muscle, and ultimately you get muscle necrosis or muscle cell death, and that contributes to ultimately what we think of as dystrophic muscle. All right. So that's the genetics and biology sort of section of this. But it helps us frame then thinking about the mechanisms that we're focusing treatment on. And I've already mentioned there's gene therapy, genetic-based therapy, there's muscle modifiers, and there's injury prevention. Okay, so we'll go through each one of these. The gene therapy and genetic-based therapy, again, just a reminder, we're going to talk about things that impact the actual DNA, but many things impact the RNA, this extra step between them. So gene therapy, no doubt, is one of the more exciting things, and I believe it was probably the first Jesse's Journey family forum in London a number of years ago, where I had the great privilege to sit side by side with Jerry Mendel, who's been a leader in gene therapy and was leading some of the very early work there. And basically gene therapy is is currently under under clinical trial development in DMD, as I'm sure many of you have heard. And gene therapy is basically saying, we know we're missing a bit of that DNA, let's replace that. Let's basically take a dystrophin gene, and I've shown it here. So this is this full-length dystrophin gene, and let's put it in a virus, yeah, for the sake of this, let's say we'll put it in a virus and we'll deliver it into the body. It's a great idea, and I think theoretically would restore dystrophin into the cells. However, the major problem with this is that full-length dystrophin is too big to be packed in some of these little viruses that we have. So what we've had to do as a scientific community is make a mini dystrophin. So you can see how much shorter this is. So it fits inside these viruses. So theoretically, if you could get that virus with the shortened dystrophin into all the muscle cells in the body, you could do it efficiently and safely, then you would have a shortened dystrophin. But these dystrophins these mini dystrophins are constructed specifically so they get the best parts of the dystrophin gene into them, so that they're the most functional. But truly, they're still an incomplete one. The second big problem with gene therapy that has come about is once you have this virus put into your body, you can't put it in again and again and again, or you have a significant immune reaction that limits the amount of DNA that's going to be delivered into your body. So these viruses are used because they go and they attach to muscle and they release this gene into the muscle. But if you see it again, your immune system says, "Oh, I know that," and as soon as you inject it through IV, the immune system comes and gets rid of it. So these are challenges that we're working to overcome, and I've put some examples of some of our partners here. Pfizer, REGENXBIO, Sarepta, Solid. There may be others, and I should pause here. My apologies to any pharmaceutical company if I've left you out. It was not in any way on purpose. So my apologies if there's, and please put up your hand and correct it if I've missed anyone. That wasn't the intention. And I've tried to do everything in alphabetical order. I'm not supporting any one company over another. So this is gene therapy. This is what gene therapy is: replacing the gene. There's another form of gene therapy as well called CRISPR. And basically what happens with CRISPR, shown this example here, is it's basically designed to come in and cut out a section of DNA. Okay, so there's natural CRISPR Cas9 mechanisms in our body that are used for DNA repair. And in this kind of therapy development, we're using those natural body things, but targeting them to certain areas of the DMD gene. So here I've just shown up top the DMD gene again, and I'm showing that in fact there's, say, 2 copies of this exon 2. Okay, and that's a mutation we see. We see duplications, and you can design these CRISPR scissors basically, and you can put them in viruses again, and put it into the body, and those then are, the goal is for them to go in and cut out this extra duplication of number 2, and when they do that, usually the DNA repairs just fine, and you restore the reading frame. Again, you can see down here this little diagram of the reading frame. So theoretically very straightforward, and theoretically could be very effective, particularly for duplications, but many mutations. And one of the main companies, there's many people working on this, but in the Duchenne space, is the Cure Rare Disease Consortium, and there is a clinical trial that's underway for this as well. Genetic-based therapies now. Let's move there. So these are largely, I've already talked about these 2 main mechanisms in genetic-based therapy: exon skipping and nonsense read-through. And there's many companies in this space. Many people who are trying to create these, I think I have a little diagram here, these little chemicals that trick the body into restoring the reading frame. Many companies feel they've got a chemistry that can go in and do this little genetic trickery. And so this is a very active space for clinical trial and for work, and no doubt the experience, and I believe we will really find good exon skipping therapies with the amount of work and dedication that's going on here. And a number of these are actually approved in the US. So Sarepta has eteplirsen and golodirsen and Camerson approved in the US. NS Pharma has a product in the US that's approved. So there are actually exon skipping drugs available commercially in the US. The other main mechanism is nonsense read-through, and the main company working in this space is PTC Therapeutics with Adlorin. And I'm not going to show examples of nonsense read-through, but I explained it a little bit earlier. It tricks the mRNA cell machinery to read through a stop sign and keep going. So I'll give you an example here of exon skipping. So in this diagram here on the far left to you, you can see that you're missing a exon 52. And so 51 doesn't line up right with 53. And so basically when the cell machinery is trying to say, oh, let's finish making dystrophin, it gets here and it says this doesn't work. And basically you end up getting no dystrophin expression, and that's expressed by someone having the muscle manifestations of DMD. But if you take one of these little chemicals, these exon skipping chemicals, which are called antisense oligonucleotides, and you block, you basically put up a signal here that blocks 51, then you can actually get 50 to interact with 53 and skip over that area and restore the reading frame. And then in fact, you do get a dystrophin protein produced. Now it's shorter. It seems paradoxical. People say, well, so you're actually making the deletion bigger to make a full-length dystrophin? Yes. Yeah, you're actually making the mutation kind of technically worse in order to restore this very valuable reading frame. This is what the cell machinery responds to. And if you can do that, you get dystrophin produced still with a gap in it, but at least you get the beginning, the end, and much of the middle. Okay, shift gears away from genetic and genetic-based things to muscle modifiers. And here, I think there's many areas that we can look at to try and enhance muscle function, even in spite of the absence of dystrophin. Some of the things you'll have heard about before are myostatin inhibition and utrophin upregulation. Both of these, unfortunately, have been disappointments in the DMD space. Theoretically, had great value, but unfortunately in clinical trials, we weren't able to see a benefit from them. But again, as I mentioned, there's many things that people are working on to try and help us understand how to take this muscle physiology that's happening and optimize it or change it in such a way that there's not so much muscle damage or degeneration. I'm going to show you one very interesting example, and that's Edgewise Pharmaceuticals. And Edgewise is currently doing a study in Becker's muscular dystrophy, planning to launch a study in DMD. But basically what Edgewise scientists saw was that the damage that happens in muscle of people with dystrophinopathies happens mostly in the fast muscle fibers, not in the slow muscle fibers. You can see that on this picture. And so, and you can measure that through disrupted muscle fibers. And you can see that in some of the chemicals that are released from those muscles like CK and fast troponin 1. You can see the elevation in fast muscle fibers showing damage, whereas in slow muscle fibers, you don't see that damage. So they said, well, maybe if we could find a chemical that inhibits the contraction of fast muscle fibers as opposed to slow muscle fibers, maybe we could slow down that damage that's happening in muscles of boys with DMD. And sure enough, this EDGE 5506 in animal models appears, you can see this graph here shows cardiac muscle and slow muscle and fast muscle, and then the amount of metabolic activity in those. And you can see that if you treat those cells with this medication, the fast muscle fibers are, their metabolic activity, their activity is knocked down, and yet cardiac and slow skeletal muscle is preserved. So this will be an interesting therapy option, emerging treatment to follow over the next couple years as clinical trials unfold. Does the inhibition of these fast muscle fibers that appear to be driving so much of the damage, does that impact the long-term profile of DMD? The next is injury prevention, and I've shown a muscle fiber here with, one of the hallmarks of a dystrophin muscle is inflammatory reaction. The muscle breaks down, the body wants to try and repair that, so there's inflammation in the muscle, and that's at least theoretically, why steroids work so well is to try and clear up this inflammation. And there are, when the inflammation happens and the muscle starts to break down, there's fibrous tissue or scar tissue basically that enters the muscle. There's fat tissue that enters the muscle. And so there are companies looking at how to try and settle that whole inflammatory cascade down. And one of them we've actually talked about already today. You've seen vamorolone come up on a number of occasions, and I won't go into it in great detail, but this is a steroid substitute that actually works to really, as opposed to prednisone, which we've heard so much about already, that settles inflammation down through one pathway, but it also affects many other pathways, hormonal pathways, other steroid pathways in the body. Vamorolone is very specific to just repressing the inflammatory pathway, and so it'll be very helpful to see how vamorolone performs in larger clinical trials. Very good success already in early phase clinical trials, very promising, not only to mimic the effects of steroids, which we know work so well, but avoid all those potential side effects. And I think you'll hear more about vamorolone from Santhera tomorrow. And this is a nice story in the sense that, the studies in vamorolone have been a lot done in Canada, certainly the current study here, and that's exciting for us to contribute to that new knowledge. So then once we talk about all the mechanisms, we need to just recognize that drug effectiveness also is driven by the pharmacology of the drug, things like side effects. How, we may have a fantastic treatment, but if it has significant side effects or problems, it may not be the most effective thing. And then patient preferences. So just talk a little bit about each of those with one example. The pharmacology of a drug is really about the concentration that you can get into the body, so how you give it, then how well it gets into the muscle, and then the efficiency of that action. So you may have a drug that works very well in a mouse model or in a cell model, But if it doesn't get into the muscle in the human, then obviously it's not going to be very effective. And we worked very closely with a company called Wave, and we still do, on a new generation of exon skipping drugs. But this trial was run in Canada. Unfortunately, we got many boys through there. We could get good concentration in the body, but when we did the muscle biopsies, despite sort of promising effect in animal models, we just weren't getting muscle tissue penetration. And so this suvodirsen was, it's not in clinical development anymore because of this. Side effects really are a significant problem in the DMD space for sure, because so many of these treatments are in development and they're new therapeutics, first time being used in boys. There are both expected and unexpected side effects that can limit how useful they are. And so again, this is downstream of the actual effect, and these graphs come from a study that we've been involved with Sarepta, which show with the 5051 product. So this is an exon skipper that skips exon 51, is sort of similar to the example we showed. We could get very good exon skipping, and we could get a very good dystrophin production. In fact, quite dramatic compared to what we've seen in other studies of exon skipping. But unfortunately, this study had the unexpected side effect of low magnesium. And so this study was held for the better part of a year, probably over a year, while we had to sort this out. And that was a heartbreak for everyone who was involved in the study. You can imagine being on a clinical trial showing effectiveness in exon skipping and dystrophin production, and then having to sit and not get that drug for a year while we worked out a safety plan with the FDA. And so these are very real realities of being involved in clinical trials, and it's not an easy business. I applaud. In fact, I'm inspired every day by the kids and families we work with, and their resilience to go through these clinical trials. And there are many examples. I won't go into it, but I do think we need to just pause. I think we need to recognize that there have, been death of boys and men in gene therapy trials. And in the most recent CRISPR trial, many of you will know that the first man who went into that trial has died recently. We don't know the details of that, but this is tough work, and it's a lot of sacrifice and risk, and we need to work very carefully together as we navigate through this, because we need to find therapies. So I just want to acknowledge that, and that we understand the risks. I'm always optimistic and hopeful, and so I don't put this up to discourage that, but only to really say that we all have to work together very carefully in the DMD space to realize these therapies. These images come from some of our boys who were involved in the drisapersen study many years ago, one of the first exon skipping trials, and this really, the injections of these really affected the skin, and many of these boys are still dealing with this. So it's tough work, and we should be very grateful to the boys who are going into clinical trial activities. The effectiveness also has to do with preferences, and I'm not going to get into this. We've talked enough about sort of shared decision making, but really understanding your values, your experiences, your preferences when you are thinking about clinical trial involvement. So Again, right now there's no approved therapy in Canada, but Canada is a very active clinical trial space. We have many clinical trials in many areas across the country, and the next thing we're going to talk about is the infrastructure and how we're all working to try and make sure people have opportunities to clinical trials if they want them. Canada has done an excellent job to organize itself. In fact, when I started in this field about 20 years ago and started to try and pull people together and say we've got to get organized, I could never imagine we would be here today. And now we have this fantastic organization called NMD4C that pulls us all together to really work on all aspects of different neuromuscular diseases, but particularly DMD, as it's the most common. And one of the platforms of this network is clinical trial work, and that's the working group that I lead and that Bonnie and Nicola are involved with, and we meet every couple of weeks or every month and try and check off these boxes of things that we need to do to make sure that we have a country, clinics, community where we can provide clinical trial opportunities to families that want to do this. And so I'm going to stop here because this is the part that I want to hand over to Bonnie. Bonnie is one of the tools or the facilitators in our clinical trial supports, and Bonnie's going to talk about her role as a clinical trial concierge. So I'm going to pause there, and we'll get Bonnie's slides up. Yeah. Yeah, just hit here.
Oh, okay,
yeah,
gotcha.
That's
Okay.
easy.
Right.
While Bonnie's getting her slides up, I just want to say thank you
So much, Dr. Oops, oh, geez, sorry. I just want to say thank you so much, Dr. Campbell. It's a lot of information and a lot of technical information, and you did a really excellent job of putting that all together and helping it make a lot of sense for us. So thank you so much for that.
Yeah. I just want to start out again by acknowledging Defeat Duchenne and Dr. Campbell for including this, the concierge on our agenda for today. And yes, it's my second job besides shared decision-making. So it comes in handy and works out well. But it is with NND4C, this position. And so I guess I have to do this, right? So you can see that what the concierge is for is more of a information broker across Canada with all the investigators, with the families, with sponsors, about information about trials. And the position works to strengthen the landscape, the trial landscape across Canada amongst all of you, families, investigators, sponsors, and probably other stakeholders, which could be politicians or education sites, and respond to the parents and the caregivers with your questions around trials that you might be interested in doing or in being involved in, and the concierge can help navigate that by giving you more information and putting you in touch with the sites that may be doing a study that would be a fit for your child. It's important that everybody understand that the position is independently objective and designed to serve as an impartial resource. So it's not funded by industry, and it is all for neuromuscular stakeholders. This slide will just show you the benefits. I've outlined that there are 4 sections here, and of course patients and families are in the center, and it's, as I mentioned, that it is to assist you with appropriate contacts, appropriate trials, if I can assist you with inclusion-exclusion criteria. I work very closely with Muscular Dystrophy Canada and Defeat Duchenne. We all work as partners on this, because if I don't have the necessary information, I may contact one of my partners, or they, and vice versa, they contact me. And I like to trial families to appropriate study sites. and many families do call and want to know, this is what my situation is. Are there trials? I heard that, for example, I'll pick on Dr. Campbell. I heard that there was the London site is doing this trial, and I'm part of their information, their research coordinators when they meet on a monthly basis. I'm part of that, so I get the information firsthand about what's happening in that trial and whether it's continuing and not continuing and the reasons why. Also, for industry and sponsors who are here today, I know it's providing you with feasibility information. I can facilitate contacts with trial sites, key opinion leaders. We can work together more effectively, I think, and provide information on both the pediatric and adult sites. It's not just for peds, it's for adults as well. It's the entire NMD4C. And for investigators, I'll go to investigators at the next slide, but other stakeholders, as I mentioned, could be politicians, they could be universities, basic scientists, people who are calling just for information on what's going on. And we also do, if you are a sponsor or an industry who want to know more about the landscape, we can arrange an introductory meeting with the leads to give you more of a broad view of the landscape and how we operate. I wanted to share a slide about what the impact of the Concierge has been since it's been in place for almost 2 years now, certainly funded through the grant that they have. And it's done quite a bit. I'm not really sure on how much you know about all the various intricacies, such as this CNDR, and the sponsors and the investigators would understand that. But it has made a big difference in impacting, broadening the understanding of how we work, and putting us more on the map in terms of trying to bring all the investigators and all the sites together so that we have a cohesive group that we're communicating with. It's been a while, and it's certainly a journey, and we've got a long way to go yet, but a lot of these pieces are being pulled together. We are looking at, we've done a bit of a metrics exploration where we're examining how long it takes to get trials studied and started, and is there a difference between sites across Canada. And we're just going to probably be doing some analysis on the data that we've collected, because we do know that sometimes there is a difference between sites, and we're working towards trying to unify ethics approvals and budgets and that kind of thing, like the timeline, so that we can get trials started, get recruitment started, and launch the trial. So I won't go into detail of some of these, but just to let you know, there's been a fair bit of impact. This here is really a map that we put together for investigators, and you can see on the lower left-hand corner, it is for various stages. If the site, the investigators are forecasting a trial, they can contact the Concierge for assistance with if they need assistance with feasibility studies, if they need assistance with, they think they're going to need assistance with recruitment, and just communicating the forecasting of that trial. They can contact me, and I can work with them to assist them in what they need ahead of time. And then once the trial, as you move up the ladder, once the trial is approved, then I can assist them, especially the multi-sites. I try to pull the multi-sites together to get a sense of where each site is at, and try to communicate with each one where they're at in their trial partway through the study once it gets launched. And we can communicate that out to the rest of Canada through newsletters and information. There is a Concierge website. On the website for Neuromuscular, there is a Concierge section, and it identifies all the trials that the Concierge supports, both adults and pediatrics. Not every trial is supported by the Concierge, but there's quite a few there that are supported, and can give you information on the status of that trial, because I do support, or the Concierge does support that trial. And then as you go further up the ladder, we can identify whether that was a successful trial or not a successful trial, what was the good news and the bad news of that, and writing a report so that everyone has that information of how it went and was it good or not so good, sort of the results and looking at the lessons learned from that trial and what would we avoid the next time and how would we make it better. And this is just a map of Canada. You all probably know this, and that shows you all the sites within the across Canada, both adults and it's probably small to read. Doesn't look too bad there from there. All the adults and the pediatric sites across Canada. So I just put that in so you can see how huge this is and how many studies and investigations take place, or studies or clinical trials can take place across Canada, and there's quite a few. That's it. Any questions at this point? Very quick overview. And Nicola is going to talk about an excellent decision aid, and that they've defeat Duchenne, I keep calling it Jesse's, defeat Duchenne has pulled together and produced and created, and it's really quite excellent.
Thank you, Bonnie, and thank you, everyone. So how much time do we have left, Rick? Are we pushing? 5 to 5, okay, all right. Perry, I think you may need to come in here and do something here to get us going. So I think I'm the last person to go. So thank you, everyone, for staying on. This is the last presentation, and I'll be quite short. And I really just wanted to take another opportunity to show you our clinical trial finder tool. And this is a tool that we really felt that our community could use, something that it's made by us, it's made by parents, it's made by individuals who are intimately involved with Duchenne, who have been through the process of clinical trials. We wanted to develop something that was easy for you to use, easy for you to understand, and also very specific to what, oh, sorry. Apparently I'm having some major difficulties with mic today. I think I've made all the wrong mistakes when it comes to mics. Anyway, and so I just wanted to recognize the individuals who have contributed to creating this tool. And Rebecca Lee, I think a lot of you know Rebecca. She's been a friend of Jesse's Journey, Defeat Duchenne Canada, for many, many years. And she's a sibling, so her brother is a young adult living with Duchenne. And we have our Rochelle, who's our marketing expert for the organization, who's done a fabulous job of really helping us with the communications and getting this out into the public domain so people are aware of this. But most importantly, Lumio, who is the organization that we worked with to help develop this tool, have been just fantastic, because they really did let us create this, and they listened to everything, all our feedback, and really worked hard to try to make this tool something that can work for you. I'm not going to go into too much detail around this, because Craig did a really great job of talking about sort of the roles that clinical trials do play for us. You know, it offers a lot of hope for our community. It gives us a feeling that we're doing something to help our children and our sons who have Duchenne. There's altruism there as well, in the fact that we are helping to contribute to the science and help other Duchenne families in the long term. And let's be real, a lot of times clinical trials, it really does mean early access to some medications that we may not see. And we see that in Canada. We're going to be talking a lot about advocacy tomorrow, so we'll get into more about that. But it is a little bit, there's a balance, right? There's the risks, and then, there's the benefits, and then there's the risks. And clinical trials, it's not something that should be taken very lightly. To make a decision to get involved in a clinical trial, you know, there's a lot that goes into that, and sometimes it can result in a disappointment. So just because there's a clinical trial that might be something that your son could qualify for, it doesn't necessarily mean that you might get into that trial, and often families might be met with some disappointment. A good example of that is, for example, some of the gene therapies, and you have to undergo testing to make sure you don't have antibodies to the treatment. Otherwise, you wouldn't be able to get the treatment, or well, because it wouldn't work for you. It can be a huge burden on families. There's a lot of work-life balance that goes into accommodating for clinical trials. Often there's a lot of travel involved in getting when you get involved in clinical trials. And so these are all aspects that you really have to think about when you're making a decision around the clinical trials. We talked a little bit about side effects. There's sort of the unknown of what that might be. And I just wanted to sort of give everyone a reminder too that, clinical trials are experimental drugs. These are drugs that are experimental. We don't know if they work, because that's why we're testing them, to see if they do work. And we also don't know if, if there may be some unknown risks associated with it too. So it's all about balancing that risk benefit. And we have a really great decision coach who I know can help make that decision when the time is right, if it's something that that you are thinking about. The other piece I also want to point out too is inclusion criteria for clinical trials can be very restrictive. And we're very aware as a community that in the adult population, there are far less opportunities for clinical trials. That's something as an organization we continue to advocate for, to broaden inclusion criteria to be as inclusive as we possibly can. There's reasons why these clinical trials have inclusion criteria, but it's conversations that we're continuously having. So we recognize that as well. And just a couple ways that we're supporting the families is developing this tool. And one of the things I want to say is this tool was really, truly developed, and what makes this tool very unique to anything else is that it's actually tailored to you. So if you, and Dr. Campbell mentioned this earlier in his first talk about, knowing what your genetic mutation is, right? Having access to that genetic report, really important, because your mutation can matter, especially when it comes to clinical trials, whether or not you might qualify for clinical trials. And so so you're able to input what your mutation is into the trial so that you actually do filter trials that are specific for you. And the point of this too is that, and hopefully we have also, the information that is available to you is information that we have tried to rewrite so that it makes a little bit more sense. Because if anyone has gone into clinicaltrials.gov, it's not the easiest tool to navigate, and oftentimes the information in there, it's hard to understand. So we've tried to take that information and sort of give it in a way that will hopefully is easier for you to understand. And finally, I just want to say that because you might see a clinical trial on here that you might qualify for, again, doesn't necessarily mean that you will get into the clinical trial. There is a process that's involved in that. The point of this tool, though, is to make sure that you have information available to you so that when you go in and talk to your clinician or your physician, that you have information that you can share with them to have an informed conversation about whether or not clinical trial or a certain clinical trial is the right fit for you and your family. And that's really the purpose of this tool. And so, oops.
Thank you.
No, I'm good.
You good? Oh.
Yeah, I think so. Yes. Okay. So, thank you. So I'm just going to quickly go through a run through, and I'm going to have to take these glasses off to do that. Let me just, make this a little smaller so you can see. And so when you go, so this is actually on our website. So if you go into our Defeat Duchenne Canada website, you go into Living with Duchenne Research Clinical Trials, there is a direct link to the clinical trial finder tool, which is this web page here. And you can link to the tool here, or you can just type in defeatduchenne.ca/clinical trials, and that will get you there as well. So this is the landing page for the tool. And I just want you to know that the information, so there is obviously personalized information that you're putting into this tool. I just want you to know that that information is not stored in any way, and it's not used
for any other purpose other than to filter what the search engine is doing for you. Once you're out of that tool, that information is gone. So it's not getting stored anywhere. It's not getting kept anywhere. So when you go into the first landing page for the tool, you actually have the opportunity to then start to put in some of your information. So let's say, for example, we'll say there you have Duchenne, you're a male, your birth date was January, we'll just say for simplicity, 2012. So you're a 10-year-old with Duchenne, you're still able to walk, and you're on a stable dose of corticosteroids. Okay. And you go into, now the next part is around your mutation. So if you know what your mutation is, you can go ahead and put it in here, whether it's a deletion or a duplication. If it's a deletion, you can actually put in exactly what the deletion is. So it might be exons, for example, 47 to 50. If you don't know your deletion, or sorry, you don't know your mutation, that's okay too. You can just say, I don't know, or I prefer not to say. It's still going to do the job for you. It just won't be as specific to your particular mutation. And I mean, as it stands right now, and as Dr. Campbell just mentioned, as he was going through the treatment landscape at this point in time, it would only be applicable to some of the exon skipping clinical trials and some of the gene therapy clinical trials. So this is what the landing page looks like for the tool. And on the side here, you can see there's a number of filters. And you can go ahead and filter even further down if you want. So perhaps you only want to look at clinical trials that are right now in Canada. And this is non-specific, so it could be for any mutation. Maybe you want to make sure that you're only looking at interventional clinical trials. So these are trials that are giving a particular drug or some type of an intervention. It could be physiotherapy, it could be something else, but they're actually giving something to you. Now, there's also a lot of trials are what we call placebo-controlled trials. And so you could be randomized into either the intervention or a placebo. And you also have the opportunity to filter for that. So if you don't want to deal with a placebo, you can just click on no placebo, or if you want to see what's available, you don't have to click that, but I can tell you right now, it will limit some of the trials that will come up. So if you apply the filters here, I'm just going to do this very quick. Because we didn't put in a specific mutation, you're going to see lots of different trials. So here we have Adalurin. We have some of the exon skipping therapies coming up. Juvenistat's in there, and the reason that's in there is, again, if you look at the status, so you can click on status. Right now it's clicked on recruiting, not yet recruiting, or enrolling by invitation only, and some of these trials at the moment are only enrolling by invitation. You can unclick that as well and apply your filters. So every time you do this, you have to apply your filters, and then it will bring it down to what's left, which at this point is some of the exon skipping therapies. Oops. Sorry, just clicking on something here that I shouldn't be. And so here you can see there's a number of different trials that are coming up. Vamorolone is in there as well. Now, this is, I encourage everyone to go in and play around with this. If you need any help with this, you can always contact me. I'm always willing to help families understand the clinical trials better or get in touch with clinical trials. I can certainly help you with that. I've tried to do my best in a lot of these summaries to try to describe them in a way that is digestible. For example, for this particular trial too, I've even included here which mutations are amendable to, in this particular case, exon 45 skipping. So trying to provide as much information as we possibly can so that you have the right information available that you can then go and talk to your physician about. And if it's something that you really want to pursue, it's a trial that's in Canada, you can reach out to Bonnie, who's a clinical trial concierge, and then she can actually connect you with those sites to start the screening process and have that conversation. I think that's pretty much all I had. Is there any questions, comments? I want to say, oh yeah.
My recollection of being on, the sites are listed too, right?
Yes,
Yeah, so,
yes.
so you could also just contact directly
Yes.
the site if you,
You can. So I have, and one of the things so this is a work in progress, and the way the summaries are showing up right now, it's all clumped together, which isn't the best. And I think for our second version of this, we'll try to like make that look a little bit more aesthetic. But right now, for example, this trial, it tells you which Canadian sites are enrolling. I also want to mention that this is a tool that we update in real time. So as soon as a trial has been approved by a certain site, we update our tool. So everything you're getting in here is in real time. And this is a continuously evolving landscape. So clinical trials might be starting up, might not be here today, but in 3 months or 6 months from now, it might be there. So it is something that I do encourage that you go in and check every once in a while just to see what's popping up, because it can change. And again, if you have any questions about that, let me know. And there is a direct link to clinicaltrials.gov. So if you do want to get some more information about the trial, you can go directly to clinicaltrials.gov to get some more information. If you click on the Learn More button as well, it provides a bit more information about what the eligibility criteria are. So what is the inclusion criteria? What is the exclusion criteria for that trial? And again, if you have any questions about this, please feel free to reach out. And also, if you go in and you're not seeing what you think you should be seeing, or if you have any feedback on the tool, please let me know, because I said this is something that we're continuously, it's evolving, and we want to improve it. And we made this tool specifically for you. So please provide your feedback. And that's it. Thank you. Any questions?
Any questions for any of the panelists?
Hello, it's me again. I just have to say thanks for all the good news that you shared with us. This is a really good, I'm excited. I'm full of hope to see how kind of investigation and research is over this. But I just thinking in my daughter, Paola, my daughter. She just been married 2 years ago. My question is, there are some genetic research for a careers like here?
are there?
Some genetic research for the careers?
For carriers,
Careers?
for female carriers. Yeah, certainly there are people looking to understand better how symptoms affect women who have a dystrophin mutation. At this point in time, in terms of therapy trials, I'm not aware that there's specific, like that any of these medications that I spoke of specifically that are in development would be in clinical trials for female carriers at this point in time. Yeah, most of these really are directed for boys who have Duchenne muscular dystrophy.
No,
type
and it's
type.
amazing. It's amazing to see how you guys are working.
Yeah.
I can only say thanks for all. But my concern now is my daughter, who is now married, and I try to avoid some risk for her. And that way I ask if you know that there's some research about them. Thank you so much.
Yeah, no problem. And it is an exciting and very hopeful time. I mean, I remember back to one of our first education sessions many years ago with our team that are here, here today too. And, families coming up and saying, why am I only hearing about stuff that's in mice, and monkeys? And tears streaming down her face. That mom came several years later, and I mean, now that we're actually talking about choices around clinical trial opportunities. the landscape has changed completely, and all this work by everyone and all the boys who are putting themselves forward to be involved in these clinical trials is remarkable. We are going to we are going to have breakthroughs for sure.
And I just wanted to mention too, the World Duchenne organization, I don't know if you're familiar with the organization, they actually do have a great website, and they do have a lot of information on carriers. It's a huge area that's been identified, and it actually World Duchenne Day this year, it was about females in Duchenne, and there was a huge piece on carriers, female carriers, and it's an area of research that is of interest that folks are looking into. So that will also evolve over time, and hopefully we'll start to see more research looking into carriers. So I encourage you to go and have a look. World Duchenne Organization is a great resource. I actually asked Rochelle to post on the resource site the family care guidelines, and Dr. Campbell did a fantastic job this morning of walking through the standards of care. Dr. Ward talked about aspects of care when it applies to endocrinology and bone health. We're going to hear some more tomorrow, but I encourage you to look at the family guide. And this is really a resource that was developed by World Duchenne Organization in partnership with other really great organizations like PPMD, which is Parent Project Muscular Dystrophy. And a lot of care was really taken into taking the standards of care and developing a tool for families, something that makes sense for families. So it's not a lot of medical jargon, it's a little bit more digestible for families around what you should be looking at different stages and different aspects of care. So I really encourage everyone here and online to look at that.
Families online, just want to echo a big thank you to everyone for you both in the room and for everyone that's speaking this afternoon. Just last question, I believe for Dr. Campbell, is stem cell therapy an option for DMD?
Yeah, so that is, cell therapies are definitely an area that people are working on. In fact, we've been working on that here in conjunction with Jacques Tremblay in Quebec. I think we're going to see more evolution of cell therapy type interventions, probably combined with genetic based interventions. So what I mean by that is taking muscle cells, converting them into more primitive muscle cells, changing their genetics through some of these techniques that we talked about here, particularly CRISPR, and then trying to reintroduce those back into the muscle or the body of people with Duchenne. So those definitely are in evolution. I wouldn't say they're at the stage of many of the things we talked about today necessarily, but that will be definitely on the horizon for sure.